What we want to do today is do this. And I think there is a context of thinking a little bit about what you might have seen centrifuges are used for. You've seen a little bit on the microscopy front. And one of the things to think a little bit about is most of the time, the samples that you were looking at were quite dilute. I mean, even the stuff that you pulled it into the water, any diagnostic sample itself ends up being dilute of the pathogens that you're interested in. So there's a common technique that's called centrifugation. And I'm assuming some of you have actually used the centrifuge. Yes? No? Has everybody used the centrifuge? Maybe? No? Okay, so I think, yeah, maybe we'll try spotting. There's one sitting right there, for example. So the purpose for today is just think a little bit about the challenge itself, and then I'll tell you two of the solutions that we have built. But then again, you should just be thinking about this challenge more broadly itself. So here is the thread that we got started on, first of all. So this would be one example. I have to share screen, right? Yes. Okay, so I think this was sort of a setting that at least I stumbled upon this problem. This is one example of a centrifuge, a heavy motor that drives and spins something really, really fast. That's what the word centrifugation would mean. And if you have any density differences in the things that you care about, they would potentially start separating out. So one example for that is what is called a QBC test, which I'm wondering, I should have picked that up. A QBC test, okay, I'll pull out a picture of it. It's a malaria test where this is regularly used. How do we get out of it? Does anybody know? If I hit escape. Yeah, I'm just trying to switch the screen, but okay, so the thread that I want to show you guys is called the QBC test malaria. So this is the challenge that we first started with. So if you look at this picture right here, if we take a drop of blood, which we'll try doing today, do you know where we left the capillaries? In one of these boxes? Oh, right here. Yeah. So you can take out a person's blood in a single drop in a capillary, right? We did that last time when we were making smears. We were doing single drop of blood. It turns out blood is made of very complex number of things. There is RBC that you can see that you're just most commonly aware of. But then you start seeing all of these other layers. I'm going to find a better photograph for that, for example. Let's see if there's so this is an actual picture. Let's see. Because there are multiple components of blood, if you could separate them, you would have a better chance of actually detecting and finding what you were looking for. And so, for example, in the context of blood itself, if you are dealing with malaria, you can see right here, the red blood cells that happen to not have the parasite versus, so you can start seeing here, these are the sets of things that have parasites. And all of these others are different stages of malaria. and they have eaten the hemoglobin a certain amount, and that has changed the cell density. But it's changed the cell density in a subtle but enough quantitative manner that if you were to spin this object, which you can see there's a little stopper here, and if you were to spin this object, many of the components of blood will actually separate out. And once that happens, then you don't have to search everywhere, which is what we are doing with octopi, right? Which we looked at the malaria smears early on, or a user, when somebody sits on a microscope, they search everywhere. You'll have to look at every cell to see whether that's malaria or not. Here, you're doing a density separation. And once you've done the density separation, if between this layer, this is called a buffy coat, and you can think about platelets are much lighter. RBCs have the highest amount of hemoglobin in it. They're the heaviest. Granulocytes, lymphocytes, this would be your white blood cells. All types of cells will start reorganizing. And they do a really clever trick. Actually, maybe if you want to now see this layer, traditionally, if I was to do it in a single capillary, the first thing that you always see is we will see blood and you will see plasma. And that's a classic anemia test, for example. So if I was to do blood plasma anemia capillary, you essentially see, and many of you might have seen this just by itself or in a capillary, that the plasma, which is the non-cellular component of blood, starts separating out. And from this ratio, you can actually tell whether somebody has anemia or not. So this is the most common anemia test that's done because you want to know how much red blood cells are there in their body compared to a given blood volume. But the challenge and the trick that QBC uses, which is quite beautiful, is everything that is in density. So if we look here, for example, between red blood cells and plasma will all be in this tiny thin layer that's called the buffy coat. This is all the other things that are present. And if I wanted to identify whether parasites were here, I needed some way of increasing the spatial resolution for it, because everything is piled up in this one tiny spot. So can you imagine a trick that allows you more resolution just in that region of density? So if you had to come up with it's a very clever idea, and I'm just curious if it falls intuitively to some of you. What we want to do is, this is density A, this is density B, this is density C, but you want higher resolution, kind of in a picture. When you want higher resolution, what do? You zoom in. So can you think of a mechanical way to zooming in? Yeah.
Take out this parts and then rotate it again.
So you could spin that again. So if you could isolate it and then run another session, one simple way of doing that is first thing if you want to stretch this in this axis, you can also use thinner and thinner capillaries, but then your centrifuges get bigger and bigger, right? Because centrifugation works on that principle where the distance from the rotation is what determines how much force that thing is experiencing. And so effectively, you could stretch it, taking it out. You could make it longer. Anything else? What else could you do if you want to stretch out the buffy coat, for example, here? And we're going to try doing something like that, but it's... So what they do in QBC, which is, it's quite clever, and it's subtly hidden in this picture, which might not be so visible, but if you look here, in the photograph right here, you can see that there's a little bobber, a plastic little piece, which has these two sharp ends that they have dropped in it inside. So there's a capillary and just a piece of plastic in it. but that piece of plastic has exactly the same density of the region where you want to stretch it. And so now the cells are actually in this thin zone between that piece of plastic and this capillary. So you have effectively stretched it. Does that make sense? The same volume that was in that tiny little thing is actually now between two coaxial cylinders. And I mean, it's again, going back to, I want to make sure I highlight ideas in frugal science. It's such a simple idea, but it's quite beautiful because now you have granularity and you can look at every one of the layers. They separate out in one shot. You don't do anything. And then you just choose the density of this specific little bobbing piece inside the capillary, and that determines which zone you want to stretch and get higher resolution at. And so I think one of the threads, this is the exact reason why you can use something like this to even determine every one of the sets of species associated with this. So anyway, this is sort of a, yes.
Just a quick question about how this works. So like,
Yeah.
is it like flush against the tube or like?
No, there is a gap. If it's totally flushed against the tube, then you will have no liquid in the middle, then it doesn't work. So it's just smaller than the tube, so it's freely could move up and down, but it will settle exactly where the density are matched.
And then like, is the point, like how does that help in doing it?
What that helps is that all of these layers that you can see, platelets, lymphocytes, granulocytes, trophozoites, all of these little layers of malaria parasite, it's very subtle in this. It's actually, there is that little triangle. You can see all of this would have collapsed into one single thin line, and you don't have the resolution to spread it. What this little bobber does is it takes, because imagine the volume of blood between this plastic piece and the outside capillary is actually very small. And so if you take that volume and you put it just in the capillary, it will all collapse to a tiny little point. So it's doing is it's just increasing your centrifugation resolution. It's a pretty clever idea. And again, it's just a simple piece of plastic. Nothing changes in the hardware. I'm just going to get some parts that hope this makes. You just cut them out. Oh, thank you so much. Awesome. Okay, we finally have the key missing material. So I'm going to take a step back now, but one of the things that I wanted to highlight is the idea that centrifugation is a very powerful, it's used in all kinds of blood analysis. When you go get a urine test, when you get majority of the blood tests, if you're getting any sets of samples that are heterogeneous, you can use centrifugation to separate them out. But the problem was most of the time when we were out in the field, we would see this as one of the field clinical condition. So we're operating in a place where there's no electricity. So although, I think this is it's a fantastic centrifuge. You have motors, it's sealed. If you don't have electricity, it's a door stopper or something else. It's not a centrifuge, right? That's the problem. And so if you have, when you're thinking a little bit about many sets of tests that require something like this, but if you cannot do that first step, then you can't proceed, or you would wait for electricity. So I think the challenge that we started in 2013 was this notion of how do we tackle centrifugation in a primary healthcare center or just at home, even in places. We recently built a COVID test out of this idea. In general, you would require, and I think what we're going to do today is, and I just realized I should have picked the yo-yo. So we got back from this trip, and I think I'm just telling you a little backstory for this, and then we'll dive in and making some of these ourselves. And the question we asked is, there are lots of spinning toys that we're all used to playing with. I mean, does anybody have a fidget spinner in their bag? No, everybody knows what a fidget spinner is, right? Yeah, at least your generation. Uh, but I mean, there are lots of spinning toys. We started with yo-yos. Then we went through basically any sets of spinning toys, spinning tops, just thinking about, are there ways to mechanically pump energy to get high enough centrifugation? That led to a lot of fun evenings in the lab. This is sort of a historic video, but it's fun to play. These are 3 generations of postdocs, all having different skills. So that's Dino right there. We first started with yo-yos. Does anybody know how fast you can throw a yo-yo? Like, what's the world record for a yo-yo throw? What would you guess? RPM? 5,000? That's actually, yeah, 4,000 something RPM, which is quite fast. So I'll let this play. This is a high-speed video of Dino throwing this. And we were making our own yo-yos and thinking about, oh, we get this transient. Could we do something in that transient? Eventually, we gave up on this idea primarily because you throw something, and I don't like the idea of throwing something with clinical samples. So also, the skill required to throw a yo-yo is much harder than you would think. I mean, some of you do that all the time, you learn, but it was just too high in terms of just skill and training. Although we did have some postdocs in the lab. This is Scott, and it turned out he was a yo-yo master. And anyway, it just so happened that some people are gifted with yo-yos. You know, that's not needed. But in that search of all kinds of toys, we then ended upon something that I played quite a lot. It's very common in India, Latin America, possibly also in Africa, because I met someone in Liberia, a kid playing with this, which we call a button on a string. And so this is what we'll be building today. You played with this as a kid, right? How many of you have actually played with this? Only you? Okay. We are maybe an older generation. I'm quite surprised. So none of you have done this at home ever before. Okay. Yeah. Oh, yeah. Simple. You can take a Coke bottle. You can take the cover of a Coke bottle. You can take a big button from a coat. We'll try, and you string 2 things in. So what's happening here? Let's just think a little bit about what was exciting is, as a mechanism, you can keep pumping energy, and you can hear it. I'll play this audio so you can hear how.
Take like 15 seconds.
You hear that sound, and that was the first indication that, oh, something very interesting is happening here. And that was the sort of the starting point of the class of mechanisms that we've now been exploring to make centrifuges. But a historic note, then we just started asking ourselves, okay, which cultures, who has played with these objects before? And this was like an incredible journey in history. We ended up finding all the way down to maybe 5,000 years ago, Jerusalem, Harappan culture, Inca culture, Mayan cultures, they've all discovered this toy independently, and they've been playing with them in Greek funerals in the olden times. You would use this as a musical instrument. So it's kind of a, you know, when you think about a sense of play, there can always be a lot of history associated with it, which is fun. But nobody in that entire time actually asked, how does it work? So that fell on us because we knew that there's historic precedence, but if we're going to now turn this idea into a project and into a product finally that solves something, we have to understand it. And that's really the gist of this entire class. You know, when you're playing with something and you have a certain idea, you truly have to just take a deep dive in understanding what you're trying to do, because without that understanding, you can't build products on top of it. You can't actually exploit the physical phenomena. So this is how it works. I'm going to walk through this just very briefly in some sense. And again, I'm stuck with that. Oh, So in terms of thinking about this, the first couple of things which we are about to build, you can see I'm drawing two strings. There is the red and the blue, and they twist together. So let's actually see if I have any functional. Adina, can you find an X-Acto knife? I think there was an X-Acto knife that, or just maybe from the supply. Do you see it? And realizing it's all tangled to a point where I need to cut them out. Okay, I'll just use this as a quick test first. So it's any shape. We don't care about the shape initially. I'm going to put a quick handle here. On one. Pen has a handle. I'm going to have you have a feel for it before. And then the other thing that I will do, because I know how fast we can make them go, is do we have eyeglasses? So I will wear safety glasses because it's always important to protect your own eyes, although kids don't do that usually. And is there a pen? You guys have a. So what's actually happening here? So we have two strings through an object, and you can see that they can just go back and forth. It slides back and forth, right? So I'm just showing that. That's what I have here. And any of you who have some buttons around, you can find a button that has two holes. Put a string in. But then if I push and pull on it, nothing happens, right? So that's where that twist comes in. So tension in a twisted string would give rise to turns, right? Because when you're increasing the set of tension, you can turn force into torque. So that's the first transfer function, because otherwise force doesn't turn into torque. I can pull as much as I want. So I'm going to load it a little bit. So what I've now done is I've turned that into twist. That's the residual twist. It has one chirality because I twisted it this way. If this has right-handed chirality, this has left-handed chirality in a mirror symmetric fashion. So you can see that the twists go in a certain way when this thing moves in a given rotation. But now if I pull on it, you notice it spins, right? But now if it spins, now it's stationary again. I mean, but that doesn't help that much if I have to keep loading. And this is where we're using inertia. So here's the trick. We want to understand the dynamics of this object. There is the torque that arises from the twist. There is a drag, which is what you're hearing. The air and the sound is all coming from the air around it, doesn't want it to spin. So you have, that's what's slowing it down. But because as you build the spin, it has enough inertia that it will keep going and it will self-twist itself in the opposite direction. So when I go here and I let go just around that time, I can let the inertia of this object go and twist it in the other way. And then I pull it again, so I can put oscillatory input in it and I never have to rewind. It always winds back up again. Does that make sense? So it's not a classic centrifuge that only spins in one direction. It's actually a centrifuge that will spin back and forth, back and forth. But does it matter? So for centrifugation, the forces are always outward. They don't care about whether you spin a centrifuge clockwise or anti-clockwise, right? So this is how that works. But one of the beautiful things about this is we can write this down and something really interesting happens. So I'm going to now try to tease apart some of them. I guess we'll make one from scratch pretty soon. I'm going to use the one with handles because if I don't have handles, it will pinch my fingers. Okay, we'll make it in a second. But the thing that I just want to get you guys excited about is this is why this is valuable when you actually dive in and really try to understand the problem that, yes.
So you said that there is no difference which way you should rotate the centrifuge. But in your pen,
Yeah.
centrifuge, there was like a ratchet mechanism to rotate one way.
Yeah, that's a second. So this is handifuge. It's a different object compared to that. And then I'll tell you why. We did that because we can in that mechanism. And actually the other part of that is there in this scenario, when you think about accelerations, the accelerations are increasing and decreasing because there is a moment of time that you actually don't see it where the centrifuge is perfectly stationary because the inertia winds it and it has to go from clockwise to anti-clockwise. So by definition, omega has to cross over the 0 point. And so what we have is an oscillatory centrifuge, but forces on all the particles that are making it move are oscillatory. And we've actually been thinking about when you have oscillatory forces, could you do new things in centrifugation that you can't do with constant forces? And I think, another thing as a challenge I'll pose you guys is we have tried certain sets of tricks that although the object moves back and forth, but can we have the disk only move in one direction? And then you can think about, oh, could there be a ratchet mechanism in the middle to begin with?
There's a special mechanism that allows you to do it.
Yeah, no, ratchets will allow you to do that. But again, we're
And two directions, when you move like this way and then the wheel here, it still continue rotating.
that's correct. Yeah, that's what we have in the handifuge. But the idea is to implement a notion like that in what we call the paperfuge, which is then there is no mechanical parts. So maybe it might have been confusing. There's actually two designs here that they are solving the same problem, but they're not the same object. This object is very different from the second object that we're making. Does that make sense? Yeah. And I think kind of, I'll show you guys a couple of videos to sort of, but then we'll just come and build and it'll internalize quite a lot. So the first thing that's important is, when we can do something like this, what this allows us to do now is to optimize the parameters of how large the length should be, how large the disk should be. And then we kept optimizing to a point where we broke the world record for fastest spinning object with human power. So at this point, the optimal version of this object, does anybody want to guess? I think you would guess the yo-yo was 5,000 RPM. What do you think? How fast can we go when we've optimized this? 6,000? What? 10? We can go 125,000. And that's when it sort of breaks your mental. So then we're not just beating this centrifuge, we're actually beating some of the large centrifuges. And again, that's why I have to think about safety. And that's why I wore the glasses is, so I think this is also another important aspect is without understanding the system, we could not have done something like that. And so the paperfuge that we will be building will be this version right now. This is what we call a very minimal version. And in this scenario, what we can do is essentially around 90 seconds, we can get complete separation of the blood phase and the plasma phase. So this would be an anemia test, for example. So for a normal person, the volume fraction of plasma to RBC is at a certain percentage. And if you have anemia, that percentage shifts quite a lot because your RBC volume is low. And so this percentage ratio between the two will decrease. And this is a classic sort of a first test. And you can see here in the plasma, there are no RBCs. Right at the layer, there is that difference. And here, this is a completely packed volume. And then the other thing that you can essentially do is now we can also do that exact same QBC tests that we had mentioned. And so here, you can really see that play out. That's the little bob in there. It's the same set of a capillary. And we can even pull out individual parasites that are then infected using imaging. So the thread is that this then goes into an imaging column to be able to detect a parasite. But then the difference between this test and then the other test is that if it's not in this zone, it doesn't exist. You can guarantee that just because we know so well about the biology and the density of these parasites. So this is what it looks like in a field condition in Madagascar. This is kind of a fun thread. This is Malagasy, so of course I don't understand it. But this is one of our field sites where we tested some of this early on. And I think it's just it's a fun moment sometimes when you sort of realize this is a village chief. And of course, we work with clinicians. The clinicians are sitting at the back, but anytime you work in a field, you also have to have permissions from the village chief and many of the other community members. So this is a fun moment in some sense, because eventually technologies that you build should also not be black boxes. If it's a black box, then nobody understands it, and then suddenly they can't build on top of it. So once we released this, something really fun happened, which is, within a couple of years, lots of other new designs. I mean, many of these are ours, but there are also all kinds of them online. And then any one of you can start building. So there are 3D printed versions of these that lock as little discs. There are large, high volume, because initially I was talking about just capillaries, so they are small volume. This is for urine-related samples where there's a much larger volume. The discs are much larger. You can see all kinds of materials. You can see those U channels and think a little bit about, I don't know if you guys see this. So that's a spiral. There's a really interesting phenomena that happens when you take centrifugation in spiraling channels, because you can see that every point you can get a very long capillary. And then again, every one of these can either be in a disposable format. So lots of fun things have been happening. And then after we posted that, there are a couple of also other tests that have come out. So inspired by that, somebody built a UTI test for urinary tract infection on fidget spinners. It's significantly slower, but it turns out for that application, that's actually good enough, which is also valuable. But again, it's very important to just sort of think about objects that are lying around us. And I think the reason fidget spinners work so well is because they have a really good bearing, and bearings are cheap. Now they are cheap. And so you can build something like that. So it would be fun if anybody wants to play with these. And then there's actually a PCR test that's been built on this platform. And one that we built for COVID recently was a slight going towards extremely infectious samples. We also wanted to build something that has enclosures and has larger and larger volumes. So this is now much more inspired from flashlights, and not the flashlight, but the recharging part of the flashlight. Gonna see if and so many of you have seen this toy before. I'll pass this around. You're pumping energy that leads to something spin. Again, all of these are much, much slower than what we can do in paperfuge. But one of the interesting things about this is then we can include a little bit of electronics. We can actually build a little detector so we can electronically test. So this one has a load cell and a Hall effect sensor. So we know exactly what RPM. So when the user is using it, if the thing turns green, that means that's the right pace to pump energy at. And then, of course, you'll see a really beautiful component here. It's the ratchet that converts, makes only unidirectional rotations, but it's actually a centrifugal ratchet, which is, if you watch it carefully, it has two other arms that themselves fling out when it's rotating. So there's a degree of freedom. So I'm just going to just first show it on video. So how do I do that? Pin. Okay, so if you look right here, this is just the flashlight, but that black piece, notice the little two knobs. At that black piece, there are these little joints. One is here at the tip of the diamond, and the other one is on the other tip, right? But you can't see that, and you should all take a high-speed video of this. What you will see is as this starts rotating, because the inertia of that joint, they fling out. And when they fling out, they engage with the gear outside and they pump energy. But now, if you rotate it the other way, they fling out the other way and then they don't engage with the gears. And that's why they can act as kind of that only, although my input here, for example, is going in both directions, right? Because this first gear is going back, forth, back, forth, back, forth. The final output gear is only going in one direction. It's not very efficient as a mechanism, but the reason to sort of broaden that out with all kinds of mechanisms is, again, for different types of tests, different things are needed. And so the one that was needed in the context of COVID was a much smaller, slower RPM. So that's 2000 RPM roughly. And I'm just going to play this video to show you guys. And then this is even a later version. And then there are a couple other versions after this that are even more stable. But now you can see we can do roughly all the way to 1 or 2 ml of samples. So the classic paperfuge can't do that. So, there we would be around 10, I would say 20 microliters, which is a drop of blood. This is much, much larger volume. And what this is a step, it's a key step in an extraction for RNA and DNA. So we've also been using this from an eDNA context. When you go out in the field, you just want to collect the DNA and not collect massive amount of sample that you bring back. So this is another very important step for doing extractions. And so if you look in here from a perspective of right here, after the sample has been inactivated, there are 2 steps in which centrifugation is essentially needed. And then, because this is COVID, we can run these assays to show that you can viably detect COVID. So I think that was sort of a background. Let's build 1 or 2 of the paperfuges. I think there's a couple of very specific supplies we will use. And then while the folks that are online, one thing I would love for you all to do is find a button and a few strings and try getting the toy working in your own hands. It can be a piece of cardboard, it can be a paper, as long as it doesn't tear. And I'll show you what we use for when we make paperfuges. Both the paper is actually tear-proof. It has the polymer coating on it. And then we put a little piece in the center that's an acrylic piece. So when the strings pull on it, it actually doesn't tear the piece of paper. So what I'm going to do is just do a construction for one to show you guys. While folks that are online, you should find any material, any piece of plastic, a cap. If you go to your fridge, I'm sure you have some plastic bottles with caps. Drill some holes in it, find some thread or a wool, and just make sure if you can actually get the oscillatory rotation going in your own hands. Okay, and then while we start, or before we start the construction, maybe Every, do you want to just introduce yourself
Sure.
and tell folks what you're working on?
Okay. Hi, everyone. I'm Avery. I'm a junior in high school in San Francisco. And I'm working on a project right now at school where I'm investigating the paper coolage to see if it can help detect postpartum hemorrhage in mothers giving birth. So if you don't know what that is already, it's when a mother bleeds excessively after delivery. And I think it's defined as over 500 milliliters of blood loss in a vaginal delivery. It's the leading cause of maternal mortality globally, but it's preventable like 80% of the time. And their mothers are dying because they're not catching it soon enough because they don't have proper screening protocols, especially in like low income and low resource places. So I thought if there's a way to detect it earlier, then clinicians can take steps to stop it. And with the paper future centrifuge device, you can measure the hematocrit in the blood, which is the ratio of the red blood cells. And if before the time, in between the time she's admitted and she gives birth, if that drops by 10% or more, then we know that she's more likely to have PBH. So yeah, I'm investigating the device and see if that's enough. Yeah.
Perfect.
Thank you for having me.
Yeah, no, and actually, maybe folks can do, let's see, this is sort of the elevator pitch time. Can folks give a one-liner of what you are all working on for Every? We can go around, see if you can tell in a single line, what is your project about? Yeah.
I'm and we're working with some of the folks as well, Smitty and Shika, on a passive deep sea sampler to collect a layer of sediment and detect the biodiversity microbial samples.
We're in this area, and we're working on replacing raspberry Pis and devices with old smartphones, so getting old smartphones and use some in all sorts of different projects.
You guys are getting pros already. That was perfect. Okay. Others are gonna have a tough one, I know. No.
The structure that allows you to make it like a widget, and if you flip it, it will be like folded in a smaller.
Yeah.
We
Emily.
on a platform.
And we're working on, we're looking for alternative options for like production that would either allow upscalability, use clothing, or for a reduction in like clothing waste and also like water pollution.
Okay, so that gives a quick kind of a summary, and I guess one of the threads is
It's good enough for us to do.
um
You want me to go the stuff in the lab? The beginning is not sticky, but.
I see, but it's double-sided, you think?
Yeah, you
Okay,
remember.
yeah, let me try this. I think if we get the regular one, yeah, let's get the regular one itself as well. So I think, yeah, one of the threads, and this is valuable Every in just some sense of the lab sessions are always open, and in terms of thinking about there's lots of people that take the class that are just not even here, which are all those folks up there. And so one of the threads would be is if this turns out to be the right kind of a solution to think about it, then what would be valuable is to kind of put together some kind of a Notion page that describes the kind of a challenge. There's lots of fun people that you can recruit from the teams itself. There's just lots of resources associated with it to make sure that you feel that you're not working alone on a problem, which is kind of the whole point of this class, and is much more about a community. Okay, so I'm going to do something here where I already have this pre-cut piece of paper. I have given double-sided tape, and I have this one little piece, which is that acrylic piece that I was showing before. And I'm going to then thread that through here and put the. So right now I'm making a device which we just call is for demonstration purposes. And since you will actually be using it in a project, then I'll kind of walk through. There's a difference between the one that we use primarily for demonstration versus the stuff that we actually use with blood. There's all kinds of safety features, and this has been one of the another important things to remember, that one thread is getting a sense of an idea. It's another to then build the kind of safety features that are needed. So there's a couple of things that are built in. There's a closing chamber. There's a very particular kind of sealant we use for blood. And this is the standard clay that freezes to then make sure that the capillary is sealed, that even if anything happens, the capillary cannot leak. And then the third thing that we actually do is we use plastic capillaries because then there is no way that they also shatter. And this is the same sets of things that are also done in centrifugation itself. And it's valuable to, and I think initially I'm assuming at your high school, you don't have access to blood, right?
No.
Yeah, so I think I would also say, don't start with blood because that itself requires, just working in blood by itself requires to be in BSL-2 because human blood has viruses. I mean, AIDS is one of them and other viruses. So even, we all get hurt all the time. We put a Band-Aid, we throw that away. That's one thing. But when you're doing research, kind of so nobody, although I poked myself to do that demo last time, you guys shouldn't be doing that. Unless you're then working in the lab, you have a BSL-2 clearance, we get the protocol, and all of that is feasible and possible, but it's important to do it right. Yeah, so I think it's I think maybe you guys will, that one thread is thinking about pathogens and other threads, but other than that, at least in the class, there's nobody that has any pathogens associated with the projects yet, right? So far. Yeah, I think you guys were one that I thought about, but nobody else, right? Huh? Baba, that's true. Yeah, but Baba already works in our lab, so he's a grad student, so he's in the lab all the time. He's I think just in general, this is a note for all the frugalists online, is that there is a level of discussion and safety and planning that happens before you pick up something when you're directly working with pathogens. So sorry, I'm talking, but I'm also just finishing building a paperfuge. So what I've done now is I just have any piece of a rod. We like this because otherwise your fingers can get pinched because these things, so it gives you a nice little handle. I'm going to, and then this piece is important because this makes sure that the paper does not rip. And I probably need another double-sided tape here because this tape might not be sufficient. And then, of course, one of the key threads in exploring for the fastest ones. Oh, perfect. Why don't I do this? We'll do this as an experiment. I have this other double-sided tape that I've never tested, and I'm going to test it to see whether it fails, and it'll give us the actual experiment. I know that many other tapes that we had used did not have the strength because it is spinning quite fast to handle, and so I think this will give us a nice example of that you have to do failure testing essentially for any device that you're working on. I don't know if anybody online, have you guys figured out, anybody has buttons spinning already? I'm also curious culturally, what do different cultures call these toys? So, in many cultures, there are different names associated with objects. Like I know in Spanish, it's called run run. Huh? Do you know what you call it?
We didn't actually-
You don't have a name for it?
just-
Spinner? Yeah, yeah. Does anybody know in India what this is called?
In Marathi, we call it as firki.
What do you say?
Firki, firki. In
Oh,
Marathi, we call it as firki.
Yeah, firki. Firki means something that's spinning really fast. It's okay, so almost.
Made from the bottle cap,
Yeah, I think the bottle caps are the best.
and there is like a special tape around the bottleneck.
Uh-huh.
We took it out, and flip it and put it on the around the cap,
Uh-huh.
and put like a pencil.
Yeah, yeah, and that part is very scary. So does everybody get what Adil just said? You should maybe explain it just one more time. I mean, I know what you're saying, but I don't think people realize. Yeah, you want to just draw it over there?
Do you have a bottle? Plastic bottle, no? No, I think. Yeah, but.
Yeah, I mean, this is supposed to be a lab session, so you guys should walk around.
Yeah.
You can come close by here.
There's like a kind of a belt around the neck. You take it out, flip it, and put it around this around the cap. And there's like a teeth around inside this band tape, like I don't know. So when you like spin it, so it becomes like a very sharp saw.
Like a
Yeah.
chainsaw.
So what we used to do with Coke bottles and others, now that's really bad, is that you would fight with your friend while this thing is spinning, trying to cut their spinning top. And that goes back to, I think in Japan, it's a very, you guys know that game where the spinning tops fight each other,
Oh, yeah.
like on a boat. What is it called? Yeah, so that's, yeah, we do that. But I mean, literally, you can see sparks fly in your hands, and it's not a good idea.
Yeah, we had a lot of dangerous games.
Yeah. Yes, of course. Okay, so this is test number 1. I have a known disk. I put handles now on, and I'm gonna spin it. So this is just first loading, and I can already feel that the weight is a little too low because the inertia matters, and I can sense that the and so now I've loaded it, and I'm just gonna pull. So can you guys hear it?
Yeah.
And already now the tape failed, but that's okay. And so at some point of time, because my spacer has failed, it will crack. And there are times where if I use the wrong kind of paper, I can spin it so fast that it will fracture. So like the paper will shatter. Remember we talked about so I'll now pass it to, so I'll just show it. There's a rhythm to it in getting it working, and it's quite fun when it tells you when to let in. So hold it here, and so there's a frequency that we're running it at. Yeah, so you have to get,
Oh, yeah.
you see, it pulls you in a little bit just at the right time. But if you don't get the frequency right, then you can't pump it up because you want to make sure. And then it's not about the strength, it's a dynamical system. So you're driving it at just the right frequency. And you can pull it hard and you will see a harder push. And if you hold like that, But this gives you a good grip. But if you keep pulling it and you go beyond a certain point, then you are suppressing inertia. So it's a tension that has to change back and forth. So you should just pass this to other people. So, yeah, and it doesn't have to remain in continuous. You can stop it. Yeah, so let's see if you figure that out. Okay, so you have to see, that's the trick. So now we have lost the turn and now we can't, but all you do is just start again. And it's, I mean, just play with it. There's nothing else other than just play. So I just get started. I pull, but then I make it loose quickly. So it gets the chance to unwind the other way. Okay, there you go.
Is
Yeah.
always bobbing like down? Like, does it ever get past, I don't know, like this line? Like, it never goes up.
No, there's no tensions in the other direction.
Yeah.
And I see that the pin has come off, so I'm gonna now make another one. While you should try teaching others to make sure that you can get that up and running. Does anybody else has the button working? Are you guys even attempting? Nobody's attempting, looks like. It shouldn't be hard to find a button and some strings. It is quite worth it.
I got one, but but I can't figure out the spinning element to
Okay,
it. How
so-
much do you spin?
You first load. So I'll let you guys now figure out. Let's see how long it takes for you guys to figure this out while I make the second one.
Have you ever made a really long one and tried to have two people link up?
We've done those calculations to show what does the length and frequency and the disk size, how does that vary with the total length? And one of the threads is actually non-intuitive, but if the lengths are quite long, there's something very special happens in the shorter strings. When the thing wants to twist, but you have run out of length, it does what a DNA does. It's called supercoiling. It forms a structure. So if you look carefully at the structures, it's not just the structure of a helix. No, I want you to figure it out.
Wanna try?
Yeah, no, there's no try. It's like you have to figure it out. It's just, you first load it. Yeah, what is it called? You do or you don't do. There's no try. Yes, yes. And then, so while we're making that, now I'm gonna make one that has the capacity to load samples, for example. So we're gonna attach two more pieces, which are these pieces that will block the capillary. So you see, it doesn't have any twist right now. So no matter how much you pill, it won't turn. So why don't you guys figure out and show?
It's like that it does it by the first one, but then it stops.
Yes, and the reason it stops is that you're not giving, you have to increase the tension and reduce the tension. Yeah, so cell phones, you can point your high-speed version of your phone on it while it's spinning and try see if you can detect. So make a little mark on it and try recording if you can collect the RPM on just that itself. Because all of you have high-speed cameras in your phone. Yeah, I think that other tape definitely crashed.
I think we can make this.
They're like benchmarking.
And then I think there are more discs, so people should come in and make their own. You should definitely make, so you can take one home. And I just want to check whether this tape is the right tape or not. So you can come on that side. Anybody else who wants to make it, there is enough supplies to make. Are we out of time? We still have 5 minutes. Yeah, so there are 2 more discs. You guys should make one. together. You start with a piece of tape, you take one of these. And when you're using tape, try not to touch it with your hands, because otherwise it reduces.
Yeah.
Try not to.
This one. Yeah, exactly, exactly. Exactly, yeah.
But that's, we don't even, just a regular cell phone would be good enough, but actually the highest ones are spinning so fast that
Yeah,
you
that's
would
what we were
not get
discussing.
you won't get the. But just test first with what is there. I'm surprised nobody's figured that out. Okay, I'm gonna put this in the final exam. Before you give your project presentations, you wait. Or maybe the trick is you get that many minutes.
Yeah,
If your partner can keep that going,
The whole presentation.
that's it. That gives you the minute. It's like the juggling.
First exam.
All right, okay, so
Yes.
point a high-speed camera to it with your cell phone and see. And then I use this to drill a hole, and then you wanna take,
Let's hold it still, yeah, there we go.
you know, maybe that big is good enough string.
5% activity.
Right there, which is then it's gonna double. It should feel just roughly that much of a string.
That's okay.
Do that.
Yes. Time lapse.
and go follow that.
Oh, it fell off.
Yeah, so I think one of the things is that this tape is not good.
Yeah. I think it's a big, I think it's a little tight.
This is not only
I started mine.
not touching it on the surface, because that's one of
Before, I think.
the things that does happen, that if the tape has been touched,
Oh, my, it's too
So,
fast. Yeah.
now you're doing, literally, you're building a scientific instrument, so you can pull in the tape, put it here and cut off the readings that you don't need, but it's to be around.
The slow can't keep pushing
Fine, that-
Then
But that's the point. So,
reaching speeds unfathomable to the human eye.
put a slow-mo on the slow, like double ear the slow-mo cameras.
Does that, yeah, would that work?
I
feel like.
that was fun.
Oh.
But maybe a little longer.
I see.
Because you want, when it's folded, it's gonna be, yeah, I think, yeah, that's actually. And I need more handles.
This worked so well earlier.
So handles. Oh, yeah.
Maybe if
Yeah, I just broke it. Let me just borrow this, I'll just cut these handles. for 1 second.
Yeah, then maybe a 240 is my max.
Feel bad.
So can any of you imagine a trick
Bad, but still
to
good.
figure out how to measure the RPM? If your phones can't do it, how would? I mean, people figured out the speed of light when we didn't. So you got to be able to figure that out. How would you do it if you don't have a high-speed camera?
Use math.
Use math, exactly. Use math.
It's actually.
No, I mean, yeah, but what else can you do? What can you make?
What happened was, did you notice that like?
No, you have to make one,
Eclipse timing
like
for-
before it existed. There was
Someone's.
clearly a moment. I mean, the light was actually a hint. If somebody could figure out the speed of light, how would you figure out how this is spinning? I mean, I don't know if I posted it on Discord. Last week, there's a paper that just came out that Leonardo da Vinci figured out and calculated the gravitational constant 200 years before Newton, Galileo, anybody,
What?
just by dropping water from his waist. And of course, there was not even math available at that time, but it's just very precise observation. So what would you build? What would you build to figure out the RPM for this?
You're talking about Oh, like laser, right? Like
That's
cutting
right.
hole.
Yeah, so start with light. What could you do with light and that object? Yeah, you can,
Yeah, so like
Okay.
one tiny piece of like
reflector.
reflection for art, like for rotation.
Okay, so then what would you do? Do you want to draw it on a board somewhere? No, it's worth figuring out. If you guys, you have to exercise your brain solving these mini problems to actually solve real problems.
Another one would be like a reflected several times.
I think you guys are in the right direction. Light is the right way. But imagine if there were holes through it,
Right.
Yeah, how much
So,
technology?
what would
do you
happen?
use? Oh, I see what you're saying.
It's
no, just how much technology you have around you, which is
Right.
plenty actually.
Can we use Arduino?
Huh?
Can we use Arduino?
But it's like saying if there's a hole.
Arduino is not strong.
Yeah, see?
Instead of a.
Yeah, what would happen? What would happen? What would you get on the other side?
Like a.
You would get a flickering signal, right? So now you have flickering light. Now you have to measure that. You've converted something spinning into flashes of light. So how would you measure? You still need to find the spacing between those flashes.
Yeah, I guess you get a receiver and you're tracking time, but you have a running clock.
Could you measure like the average intensity of the light and then see if it's lower than like the where it's-
That is a very clever idea. You see, now we're talking. So this is pulse width modulation instead of measuring, because if light was coming very, very quickly, the overall intensity might actually be different. And all you're doing is measuring intensity.
Right.
And you- Yeah, that's a very clever idea. Yeah.
I don't know why. Something that I just said.
We broke it.
No?
Yeah, because there's no like.
Oh, you cracked it. Yeah, because that's because the tape was wrong. So we're gonna test that. And so suddenly, you know exactly why that piece is important.
This part.
Are you guys waiting for handles? Sorry, yeah, no, I'll just give you guys handles. Sorry, let me take the blade. PVC pipe handles or the more aesthetic. Okay, I think these are.
Maybe.
So are there any like special features to this paper that?
Yeah, this
Okay.
cannot track here even at crazy RPM.
Well, like what about it?
It has a polymer coating on it that strengthens, and even the fibers that we use here, they are also, you can see the fishing line.
Yeah,
These are designed for high strength conditions.
is this paper hard to get in these other countries?
It's a very common paper, used in all kinds of packaging and everything.
But we can use it.
You don't have to use it. We're just good, it's convenient. The reason we use this paper is we had thousands of tons of it with the full scope. The thick paper. Yeah, so let me show. So the trick, just so whenever you're starting, you first pinch the two hands and then just load the torque. So you just are loading a twist configuration and give it enough time so that you have, in the first go, you can get it started.
Also, you don't need to like pull it for too long. Just kind of give
That's,
it
if you pull it for too long, you're fighting the counter rotation. And it's about learning that frequency. So I just do that and then I bring it in. And the act of bringing it in is as important as pulling. Because if you keep pulling while it's trying to go the other way, then you are actually pumping and taking energy away from it. So in this type of a mode, you have to see, and then this is what changes with the length. That at a certain point of that length, that coordination kind of breaks down.
Oh, because it can.
No, because you have to predict when it's starting to now turn, and that's what I mean. You let this fold. These are called secondary folds. This is what happens with DNA when you twist it. You see those?
Oh,
Those
yeah.
folds are folds of folds
Ah,
right there. So this, it's a twist of a twist.
mhm.
And this is what stores a lot of energy. So in the end, in configuration wise, if this could not happen, then again, the instrument will resist. Because it's like, oh, I
Yeah.
don't have any string left. But
Yeah.
no, there is still configuration possible. And so then the turning that into much more of a device is what we do next. And I think one thing if you're ever using it, you've got to use now. This part is all good, but now since we're building a loader, then you always have to use safety glasses. So the next bit is the positioners. And the positioners, we always put equal number, and we usually just do for 2 samples, but they have to be balanced. Because if the weight on this was not balanced, what would happen? It would not be balanced. A very fast spinning object that's not balanced is actually not good. And so
Here, can you give it a try?
you should solve it. It just takes time. This is why kids get hooked to it, because
Something that
what are you trying to do?
I want to pass something so it's easier to wind. It's really light. So
Well, you have to pinch it here. And you don't need that much. Okay.
I fixed it.
I'm just building that up.
Hmm.
That's it. Now we got it.
That's confusing. Everyone started.
So there you go. Yeah. It's just in the early stages, you can just build that up. I think the length is a little too long, basically. I'll cut that. Okay, so we mounted that. We have mounted. And you can see why that is important, because we don't want that to fly off. And it has a desired.
Better actually.
And so I think when I'm gonna send you the protocols, I'll send you a very specific tape. So this is not really the perfect tape either. And then we're going to load a capillary. So let's load this, for example.
Okay.
And the capillaries that we use are the shorter capillaries. But since I don't have it, I'll just break it. This is not even like this, but if I just do the tracks of glass. Glasses.
Should be tightened a little bit here.
And this is a very sealed, so what that does is it seals. Yeah. You would only seal it after the sample has been loaded. Loaded and sealed. At this point, I'm assuming this would actually already have the sample.
Yeah.
And then, do you know, Ellie, what time the group meeting is?
We have a half hour to agree on that. I might
Wasn't
start
there-
talking about.
Yeah, actually, yeah, that would be helpful. So now we have mounted that, and I'm just gonna show folks, for a second. Oh, a two-way, I've never seen that before. But this really requires coordination with your team, so you know you're in sync. So now we have done that. And I'm just gonna do a secondary thread here. And this is the very old design. There's actually a new class of designs.
There we go, nice
on.
coordination.
Those are just the plastic polymers themselves. We use this.
Oh.
What they allow us to do
Such admirable teamwork.
with
Keep
sealing
this energy.
mechanism on top of this mechanism itself.
Super, wait, no, super.
super.
one of the
Greens. There we go.
requirements in centrifugation and other threads. are
Oh, a name
also
change,
fertilization.
Yeah.
So what that does is there's another disc that comes
Wow.
in and
Oh, do you have your little?
closes that so that this chamber becomes a double
Yeah,
chamber.
try.
And
Yeah.
That's okay. I've
that's
tried it.
actually valid in terms of
Wow, we're getting hang of it.
cooling mechanisms. So now I mounted that.
Wow, that's cool.
Now I am going to wear that. I'm gonna work it out in the-
Okay. No, you have to keep it tight. What?
And so at this point, you can start loading,
No, it's
just
fine.
load some sugar water.
That one.
And it would just
You
be
guys had an impressive run.
30 seconds.
That
And it's not about that much force that you're applying, it's not pulling that body.
No.
Phenomenal.
The frequency.
Wow, look at us.
Maybe that would be all I would need. So at this point, I would just load and then
Okay,
check
good
in
work.
whether you see the ratio already.
Here.
So I'm gonna give this to you.
Hmm.
And then the thread is, let's do that a little bit later when we don't have a class, then we can really sit down and think about, okay,
Maybe not mine.
what is the set of things that you need to get to a
Because the
point
momentum
to
should be
prove
the
that
other
this
way too,
device
right?
actually solves that
Yeah.
problem. And then
We need
if
to
you want,
increase the-
Abina and Ellie can give you access to our Discord.
Wow.
Where all the lecture notes and
also
other
not
things are.
prepared.
For the project
Yeah.
specifically, the thing that I would like you to make is this format that we call Fred Park. So let me just show that to folks.
Technically,
So just folks that are still online. So now I've added the two capillaries, and what's very important is that little stopper. And that essentially makes sure that the capillary is not flying around. Because you don't want it to fly away, that would be really bad. And then on that note, I'm also just gonna say bye. I'm sorry, today we couldn't start early on time. But again, for folks, I'm assuming everybody has a team. And then if you need one-on-one times to sit down, have discussions, please request that on the Discord. So I have an intuition for how many more people we need to sit down with. Yes, anybody who hasn't done Fred Park can do that on Tuesday. And then speaking of Fred Park, big and exciting. So this is what we do for all projects. Where would our Fred Parks be? Yeah, right there. So we go in here and now I have to choose. I'm hoping Alankrit did a good job so he did a very good job. That's good. So what you can see is we kind of use this framework in design. You always start with what are the minimum requirements that the object should fulfill to be able to meet the criteria that it is going to work for the task that is at hand. Then there is design parameters. This is the problem really in some sense. This is really the solution side of that story. And then this is the analysis that you need to be able to, what are the minimum tests that would be done to either rule out an idea? And then there are many ideas, so design parameters can have many different solutions, and you would rule out or pull one or not have the other in terms of the analysis. This is what proves to you that, oh, this was the right approach to continue going. And then, of course, every one of them has references would be your data or things that are built on top. Risks would be, what are the other factors that the whole thing doesn't work? And the countermeasure is something that, oh, and then you figured out how to counteract that risk.
Okay,
So yeah, I think try building this.
and this
Uh,
is on the Discord.
This is on Discord channel. These are for other people's projects. You can look at, there are 10, 15 of them.
Okay,
It'll also just give you the breadth of types of stuff people are doing.
okay,
Okay?
yeah, thank you.
No, absolutely. I'm sorry we started a little late, and
No
then I
worries.
will catch up online sometime.
Okay,
Yeah.
thank you so
Yeah.
much.
Yes, let's type. Say that again.